Welcome everyone. So we're just going to wait a few minutes for everyone to join. But if you want to, before we start, you can put in some introductions about yourself into the Zoom chat and then give it a couple minutes to let more people join and then we'll get started. Okay, we're just going to wait 1 minute more just to give people a couple more minutes to join. All right, so welcome everyone. Today we've got Tristram with us. He's going to be giving a talk about sexy scents and how scent influences the behavior of mammals. If you've got any questions while the talk is going, you can either put them in the chat or even easier, if you look at the bottom of your screen, there's a Q&A box. You can click in that box, put in any questions you've got. And then at the end of Tristram's talk, I'll go through and read out all your questions to him and he can answer them. So I think with that all out of the way, I'll just hand it over to you now.
Great. Well, thank you everyone for coming. I'm a behavioral biologist. My background is animal behavior. Nico Tinbergen was one of my heroes. My own experimental work on pheromones has been on insects, especially moths and beetles. But about 25 years ago, Cambridge University Press approached me to write the first synthesis on pheromones and behavior for some 25 years or so. And it was a daunting task, but it introduced me to the special world of mammals and fish and every other kind of animal. And I was amazed with what I found. What particularly surprised me was the similarities across the animal kingdom. But mammals are special, well, as you know. What I want to do in this talk is share what I've learnt about pheromones and about how mammals use these signals. There won't be much natural history because you already know that, but also there's great film on wildlife programs. I want to go into the things which don't appear so much on David Attenborough. Well, mammals are smelly, complex, intelligent, and really hard to study. And this has led to some controversies about whether they have pheromones at all. And I'll take you through some of those, including the big question about whether humans, as mammals, have pheromones. Where I want to leave you is with a deeper understanding of pheromones and other smells, the smells that animals also use to tell each other apart as individuals, and give you some criteria that you can apply to claims you see online and in the literature. Of course, what all of this work did was to introduce me to the worlds of people like David MacDonald, Pat Morris, Jane Hurst, and many others, to name just three. So this is the book that got me into animal behavior and pheromones, and it gave me a lot of pleasure. Now, what it introduced me to is all the ways that mammals use smell. They use it, of course, to find food, to avoid being food. So mice are very sensitive to the smell of cats, some of those smells that cats produce and leave around. Mammals also mark their territories. They use it to find and choose their mates. The smells I mentioned that are different between individuals are used to recognize partners and family or clan. They're used to recognize mother and offspring. So there's some mammary pheromones that we'll learn about in rabbits. And there's also recognizing mate readiness. Is the female about to ovulate? So marking behavior. This is an antelope marking a twig on its territory using its orbital scent gland. And that's actually something important about mammals and scent marking. Mammals have skin glands all over their body, and you can even distinguish some species of rodent that are closely related by the position of their skin scent glands. So scent glands can develop anywhere on the body, and there are also some internal scent glands too. This is a mara, a South American animal, a male spraying its female with urine to mark it in the herd. This is a cat marking with its chin gland. So there are lots of pheromones used by cats and dogs. And sometimes what you mark fights back, and the dog wasn't expecting that. So dogs mostly do their own spraying on fire hydrants. Now, one of the pioneers of work on mice has been Jane Hurst, of course, and this is one of her films. And what you see there is the mouse walking along its trail, and you'll see at intervals there are these bumps, which are where it's repeatedly laid down its scent marks. And the scent marks have lots of protein, and they give off their small molecule scents very slowly. But this male has to mark his territory repeatedly. He's now detected an intruder, and they sniff each other. They detect who's the intruder, who's the territory owner, and then they fight. And females also note the scent marks, and they also note the variable smells that each male has. And so they can tell when they meet the territory owner that that's the territory that belongs to him. There's a special behavior called phlegming, which gives access to the second nose. And here's a stag lifting his upper lip, drawing in the air from the female and drawing in her molecules into his second nose, the vomeronasal organ, which most mammals have, but not all. Here's a kangaroo male sniffing the cloaca of the female to detect whether she's ready to mate. Now, there's a lot of sniffing going on when mammals meet, and here are two dogs sniffing each other decorously. Now, as they sniff, they get all sorts of information, but it is of two different kinds. So with the red background, they're detecting, although if they've got their eyes open, they know it's another dog. They're detecting the species, whether it's a male or female, whether it's a puppy or an adult. And if they're sniffing a female, they're able to detect whether or not she's in estrus. But they're also getting highly variable information, and this allows them to remember this particular individual. And they can do the same, of course, when they're sniffing a lamppost. Who else has been there? It's a bit like a billboard. Now, the thing about the ones in red is that these are anonymous. Every male dog has the smell, the pheromones of a male dog. So these are kind of anonymous, and these are the pheromones. The blue smells are these highly variable smells that differ between individuals. So we've got two kinds of smells. We've got the pheromones, the same in every male, that are anonymous. And then we've got these signature mixtures that are variable subsets of molecules of an animal's chemical profile that are learnt by other animals and remembered for when they next meet. So that leaves the question, what are pheromones? Well, they're chemical signals transmitted between individuals of the same species, so it's intraspecific. The word was invented in 1959, and it comes from two Greek words, transferred excitement. So these are invisible signals, and we've known about them for a very long time. I think going back to prehistory, but we've certainly known about them since the ancient Greeks. But I'm sure when we were living in caves and making those cave paintings, we were aware of them then, as every hunter would be. The ancient Greeks knew that a female dog gives off secretions from her vulva that male dogs will follow if you put it on a towel. In the 17th century, we have the first guide, the first book about beekeeping. And there's good advice for beekeepers that if you're stung by a worker bee, you should pull out the sting because it will be pumping out what we would now call not an evil humour, but an alarm pheromone. And in 19th century France, you may have read Jean-Henri Fabre's beautiful account of the peacock moth virgin females attracting males into the house with a flick-flack of the wings as they fly around the room. Darwin was well aware of smells. In his 1871 book, The Descent of Man and Selection in Relation to Sex, he was clear that we should think about smells in just the same way as the peacock's tail. And he writes, during the season of love, a musky odour is emitted by the glands of the crocodile and pervades their haunts. But he also talks about smelly male elephants in Must. He talks about smelly goats, and we'll be returning to those later, but also pythons and a particular smelly bird in Australia. And he suggests that these smells have evolved by sexual selection by females. And he could say, not a success of the fittest, but I've said success of the smelliest. So we've known about these smells, these attractive smells, these powerful smells for a very long time. And surely these would be chemical signals. But the challenge was the chemistry of the period. The quantities were far too small to identify, tiny quantities per individual. And we had to wait until 1959, and this was after 20 years by a large team studying the domesticated silk moth, Bombyx mori, Adolf Budendant, who already had the Nobel Prize for his work on the sex steroid molecules. So what we have on the left is the female, the big bulbous female with her bottom where the pheromone is coming out, the pheromone gland, and the male is wing fluttering. He's got the big antennae to detect the pheromone. So we have the male on the left. There's the female coming in, and he starts to flutter his wings in response to detecting the pheromone. And he's gradually going to work his way towards the female. Now, silk moths can't fly, but if this was a wild species of moth, the male will be flying upwind in the dark to find the female. So what Budendant was doing was using the bioassay of the wing fluttering as the gold standard for identifying a pheromone. It starts with that bioassay. He needed to isolate the pheromone from 500,000 female moths. He then identified the molecule, synthesized it, and then went back to the males to check with them that he'd got the right molecule. And basically, these are still the essential steps for identifying a pheromone. So bioassay, isolation, identification, and then confirmation with the bioassay back again. Since 1959, pheromones have been found all across the animal kingdom, and you'll recognize lots of your favorite animals there. Pheromones work just as well underwater, and that was one of the surprises for me. So a pheromone in any animal is a chemical signal between members of the same species. The way we know that the signal has been transmitted is a behavioral or physiological response in the receiver. And what we're looking for are molecules that are the same in, for example, all males of the species, but perhaps in different amounts. There might be super males that are giving off a lot of pheromone. What is unusual, or rather, what was a surprise after the silk moth, is that in many species, it's a combination of molecules, not just a single one. And something that did surprise me as I went across the animal kingdom is that it's detected almost universally by the sense of smell, not taste. And that's certainly true for mammals. So what kinds of molecules have evolved to be pheromones? Now, it turns out there's nothing special that allows you to point to a molecule and say that's likely to be a pheromone in any particular animal as you go across the animal kingdom. It's limited only by the number of and range of molecules that organisms can produce or obtain. There are some bees in South America, orchid bees, that actually go from orchid to collect the right smell to attract their female. And in mammals especially, there's lots of fermentation. So in those sac-winged bats, there's a fermentation of urine, which the male has to squirt into this wing sac, together with secretions from the wing, scent glands, which ferment beautifully in a very attractive way to females. And he wafts that smell over them. And potentially any molecule can become a pheromone. And that's something I could talk about afterwards, if you like. What you can say is that you can relate the molecules that do evolve to be pheromones to their function. So for ant alarm pheromones, these molecules are small, volatile molecules that will disperse quickly. You don't want to be in an alarmed state all the time. Whereas if you're a hyena marking your territory for your clan, then you have large involatile molecules in these fatty secretions from anal glands. Now, the thing that revolutionized pheromone research was chromatography. And you've probably done this yourself. This is paper chromatography with a solvent moving the different dyes up the sheet of filter paper from the spot of black ink, which usually turns out to be made up of lots of different colors, hidden in that black dot. And that's paper chromatography. What really made a difference to pheromones was gas liquid chromatography, invented towards the end of the 1950s. And what we have is a mixture of molecules in the sample taken from your mammal, whether that's urine or a swab from a scent gland. That's injected into nitrogen or some other inert gas that's blowing along a column. And the different molecules take different lengths of time to reach the detector. So what we see is the mixture of 1 and 2s come out at the detector as separate peaks, 1 and 2, or however many. Now, if you look at an insect pheromone, like this woodworm, now this is another insect that has just one molecule as its female sex pheromone. Lots of moths have anything up to 7 different peaks, but that's a small number. So it's a relatively clean trace. It's level until the peak at a certain length of time in the oven, and the gas blows out this particular peak, which you can then identify in the mass spectrometer. So it's not like that for mammals. And this was realized very quickly once mammal samples were put through the GC. So not every smell is a pheromone, and mammal chemical profiles are very complex. So if we look at a human armpit, there are at least 700 molecules. There are probably thousands. And that makes it very hard to study, basically, to try and identify pheromones. Now, what I've done in the next slide is create a kind of imaginary trace. So I've gone from hydrocarbons, these very small molecules, fatty acids, um, produced, studied by the GC. And at the other end, you can use another kind of chromatography, HPLC, and large molecules can go through that, like peptides. And so the molecules are made up from many sources, secretions, there are molecules associated with the immune system, hormones affect what molecules go into that mix. I've mentioned bacterial symbionts like that sack-winged bat. What you eat affects what you smell of, and you'll know that if you've had fenugreek or some other pungent ingredients that taste great, or garlic for that matter, that taste great but come out in your sweat for ages. You might also have scent marks put on you if you're a badger. You might have been squat marked by your fellow family members, so you'll have some smells that come from them. And I've mentioned those orchid bees collecting from flowers. And you'll smell different too if you are carrying an infection. So you can imagine all those different smells, but hidden in that complex background are some of those pheromones. And what we'll find is that those pheromones are the same in individuals of the same kind, whether they're dominant males or females or subordinates or juveniles. And there's a lot of inter-individual variation in mammals. So here are two unrelated female badgers, some work done by Christina Bushing. And what you can see is their traces are completely different. Now, if there are some pheromones hidden in that, those peaks would be the same, but because they're eating slightly different things, they've got slightly different bacterial flora, they might be different ages, they've got different genetics, their overall profiles are very different. Now, you can imagine pheromone researchers looking at mammals and thinking, how on earth would you study that? And this is very different from insects with those clean traces and easy to study, relatively speaking, sets of molecules. So that led to questions in the 1970s about whether mammals could be said to have pheromones at all. So in the 1970s, a bit like art movements, there was a bit like a manifesto from pheromone researchers. Gary Beecham, Dick Doty, and colleagues wrote a statement basically saying that pheromones as a concept really shouldn't apply to mammals. Mammals were too smelly, had too many complex odors, were too intelligent, learnt too much, had behaviors that were too complex. And so really the ideas of pheromones applied to insects and lesser animals shouldn't apply to mammals. What I found fascinating is by the year 2000, Gary Beecham had actually come round to the idea that there really were pheromones. By that time, he was director of the Monell Smell Center in Philadelphia. So he changed his mind and actually started to acknowledge that mammals really do have pheromones. And they do. So it's a mixture of small and large molecules. On the left, we have the rabbit pheromone. This is the mammary pheromone we'll come back to. The pig one you're probably familiar with. This is in the boar's saliva, which causes the female pig, the sow, to go into lordosis. And mice have lots of pheromones, small molecules, but also this peptide, darsin, which was named after Darcy. And here he is coming out of the lake from the Jane Austen film. And Jane Hurst thought this would be a great name for a sexy peptide pheromone. Well, Doty remains unconvinced. And in 2010, he published this book, The Great Pheromone Myth. And I think really he creates some straw men. He argues on the basis of a misreading, I think, of the insect literature that certain things must be true for things to be pheromones. So first, molecules must be species-specific. Well, that's not true. There's a wonderful story about elephants that when the female sex pheromone that's in the urine of female elephants was analysed by a moth pheromone chemist, it turned out that the molecule was one that 140 species of moth include in their mixtures. Now, male elephants are not going to be excited by a female moth because she gives off picograms of pheromone and he's looking for gallons of female urine. And male moths are not going to be confused because the female elephant only has that one particular molecule, not the right mixture that makes the combinatorial pheromone that each different moth species needs. So different molecules can reappear in different species by convergence. Pheromones must be innate. Now, innate means that it's almost as if you're born with a particular behaviour or response. But Pat Bateson, through his long career, emphasised that what looks like an innate behaviour, because under normal development, every animal expresses that particular behaviour, whether it's cats to catnip or whatever. In fact, animals may have to be exposed to certain things as they develop as a kitten to express that behaviour later. I don't know if that's the case for catnip, but it certainly is for some things. So, for example, for a mammal's visual cortex in the brain to properly make the connections with the eyes to see an image, the eyes have to receive visual stimulation during critical periods after birth. And if it doesn't get it, if the brain doesn't go those visual signals, then the brain doesn't develop correctly. And that's true for some kinds of olfactory development too. And another idea is that the pheromone response must be invariant. Well, actually, that's not true for male hamsters. A male hamster has to have a certain amount of testosterone in his blood, otherwise the amygdala in his brain won't let through the signal. And we find that in moths too, that a male moth in some species switches off even though his antennae are receiving the pheromone. So another myth, not from Doty, but from others, was that pheromones were only detected by that special second nose. And, well, actually, that's not true. So just looking at mouse pheromones, there's a sensory overlap of the regular nose, the main olfactory epithelium, that's the nose that we have, and that special second nose that we don't have as adults. But the mouse does have it. And what you'll see there is that small molecules are detected by both the main olfactory epithelium and the VNO, but some of those peptides are only detected by the VNO. And if you go higher in the brain, you find that the inputs from those two noses are integrated in the amygdala, and it's actually a complex integrated signal that goes up to the hypothalamus that will determine what hormones are released and how the animal responds. So there isn't a clear division between the main olfactory epithelium and the VNO in terms of whether that's the area that detects pheromones. So mammals do have pheromones. How do we find them? So how do we find the candidate molecules from hundreds of peaks? So we could compare males and females. We could compare castrated males versus intact males, because testosterone is often required. We can look for peaks that differ. And there's a great example from goats. Now, I mentioned that the male goat is very smelly in the breeding season. He produces smells from the skin on the scalp. These molecules are very attractive to female goats, but also they trigger a hormonal response and trigger ovulation. So these Japanese researchers ran the secretions from the male, the head of the male, he sort of wore a shower cap effectively, ran them through the GC, and very spread out here, we're looking just at that grey section. And if we blow that up, we can compare the intact male with the castrated male. And what we can see is that there's a particular peak that's found in the intact male, but that disappears if he no longer has testosterone. And that allowed the researchers to show that 18 molecules were produced by the intact, but not by the castrated. And when they tested this as a cocktail for the female response, one of these recreated most of the activity, but fascinatingly, not all. So there are other molecules that are needed for the full effect of the natural head extract. So we can say here that we have a robust bioassay, a physiological response in this case, and we know in detail what the researchers did. Now, often we don't know all of that, so we have to have an operational definition, and I'll go through each of these in turn. So the first is that the synthetic molecule, a bit like bombicol, if we synthesize the molecule, we get the same response from the receiver. So this is the mouse, it's another Japanese group, and they're looking at a molecule that the male releases into the urine. It attracts the female, it's dependent on the androgen, the testosterone. They even found a receptor in the female that is detecting this molecule, and the synthetic molecule is just as attractive as the original. Here's that rabbit mammary pheromone, and on the right there's a rabbit pup responding to either milk or molecules from the rabbit milk on a glass rod. And what Benoit Schall showed here was that a particular molecule leads to searching behavior and seizing behavior, and that's sufficient to elicit this response from the rabbit pup. And other molecules didn't have such an effect. It's important to show that these are natural concentrations, and here we have showing that the concentration of this molecule occurs at the same concentrations as in milk, and the response curve is the same. And there's a credible path for evolving, that rabbit pups need to find a nipple fast because they only have a very short period to feed. And of course, there's huge selection pressure to compete and lock on, fighting against all your siblings. So that's enough with pheromones. I want to move on now to individual variation and remembering smells. So societies organized by smell. It's all about learning of a signature mixture, either for individual or family odors. So to recap, pheromones are the same across the species. The response is innate or hardwired. Signature mixtures are what animals learn for recognition from these highly variable chemical profiles. So most pheromones are species specific, but for recognition, it's all about difference. And here's one way, there are more examples in the book, of how this happens. So animal A meets animal B, perhaps a sibling, remembers their smell, and when they meet again, animal A recognizes B, and vice versa, because they recognize the smell. And this is true of mammal territory neighbors. It's how the pair bond works in monogamous mammals. Ground squirrels only let in family members, and they detect that by smell. So normally, you learn the smell of your siblings, but you can show this is learnt by taking unrelated animals and feeding them the same diet, and they'll learn to associate with animals who've been fed the same diet. So this was done with spiny mice, and these are unrelated animals. They're being fed the same diet, and they come to choose to associate as if they're family with animals that smell the same, that they've learnt to think of as, well, basically family. So signature mixtures are this variable subset of molecules of an animal's chemical profile. It's learnt by animals to distinguish individuals. Lobsters can do that too. And it's also used, of course, to choose not to mate with a brother or sister. So you learn who your family members are and avoid them later. And of course, it's how you know that other mongooses are part of your kin group, part of your group. And that's also true, of course, in ants. That's how ant colonies know that you're another member of the colony, not an intruder. So, using rabbit pups as an example, they learn their mother's smell, signature mixture. Every mother is different. They also respond to mammary pheromone, which is the same for every rabbit mother. And this is that molecule, and on a glass rod, they will suckle to that particular molecule. Now, why would we expect humans to have pheromones? Well, the first reason is that we're mammals, and mammals are smelly. If you go to an Icelandic swimming pool, in the showers, they have signs saying very explicitly, these are the places to wash. And basically, it's your armpits and your groin. And the reason for that is that our smells change at puberty. Those apocrine and sebaceous glands and the hair start to grow. And we start to change our smell as we go through puberty. And with any other mammal, we'd be looking for pheromones. Now, there was an idea that perhaps our sense of smell wasn't good enough. And there was an idea that humans were poor smellers. But in fact, Shepard, who also wrote books about wine tasting, argues that actually we're rather good smellers. For some molecules, we can smell almost as sensitively as a dog, so long as it's close enough to our nose. And McGann, in a later paper, said poor human olfaction is a 19th century myth. However, human pheromones, and if you've ever searched on the web for pheromones, you'll see lots of websites trying to sell you some. And it's an example of bad science. So these are the two molecules, and it's a case study of how not to establish the identity of a human pheromone. So there are two obscure steroids. And if you search up until 1990, it gets almost no mention. Neither molecule gets a mention. But since 1991, and I checked this morning, we're up to almost 1,500 papers citing these two molecules. So what happened in 1991? Well, these two enterprising professors published a paper claiming these two sexy molecules were human pheromones, and they started a corporation to sell them. But why did they choose those two molecules when, as you'll remember, there are hundreds, if not thousands, of molecules? And we actually don't know. It's quite bizarre. There is no scientific paper that really establishes that. So if we're looking for the gold standard of bioassay-led evidence, the answer is no. None of these steps were ever carried out for those two molecules. So there are now at least 65 positive studies for molecules that have no evidence of being pheromones. How did this happen? Well, studies on human pheromones are very like, well, they're basically a branch of psychology. And what we know from studies on human psychology is that you can get this situation if you have lots of false positives, underpowered studies, looking for significance, and not publishing negative studies. And so I'm afraid that's what's happened here. And there's more in the references, which you can find later. There are some other problem pheromones, and these are the commercial pheromones that are sold by vets and you can buy on the internet. They're based, like the human pheromones, on patents rather than robust scientific studies demonstrating from first principles that they are pheromones. There are lots of positive trials, but when you go back to what's the evidence that they're pheromones at all, those are simply lacking. And that's one of the sad things, because all of these animals, cats, dogs, chickens, pigs, all have genuine pheromones. It's just most of the ones that are commercially available are not scientifically based, in my opinion. There might be a human pheromone, and this is a mammary pheromone, and human babies learn their mother's smell. Every mother is different, but they also seem to respond to what might be a mammary pheromone, as yet unidentified, which seems to be the same for every mother. And it increases search behavior from the baby. This is the areola gland. This little droplet of liquid is secreted by a lactating mother. And this is work by Benoit Charles in Dijon, in France. And the response of the baby is to stick out its tongue and start to root. Now, Benoit Charles and Craig Roberts in Stirling have got a grant, and they're now looking at this in more detail. And I think this is probably one of the best candidates for a human pheromone. We've got a fantastic bioassay, and now we've got good psychologists and chemists looking for what might be a pheromone. So I'm optimistic, and let's see. Where I want to end in just a few moments is the state of mammals in 2026. And you'll be familiar with the effects of climate change and habitat loss. And you probably have seen this graphic from Our World in Data, based on a PNAS paper. And basically, what this shows is that wild mammals, those 5 dots in the bottom right-hand corner, are the total biomass of wild mammals. It's a lot of different species, and if you're an elephant shrew, you don't add up to many grams, but the point is that the overwhelming 95% of biomass is either humans or our domesticated animals. And that really is serious. We're losing habitat all the time for palm oil, for urbanization. We're losing habitat too to climate change, where animals simply can't follow the changes in habitat fast enough. So what I found myself doing was becoming an activist scientist. So this is me outside News International complaining that Rupert Murdoch's Sun and the Times really don't discuss what's happening in ways that make it understood for all of us, what's happening both to our mammals, but also ourselves in terms of coming food shortages and all the things that you may be aware of. Now, something that's really current at the moment is a campaign for a national emergency briefing. And the idea is that what we want is for the government to have something like the kind of briefing that went out on all television stations during the time of COVID, explaining about the virus and what we need to do, and basically how serious things are. And one of the talks is by Paul Behrens, who works at Oxford on food and diet. And one of the things is we perhaps need to look after our wild mammals better and move a bit more towards a balanced and partially plant-based diet. So that's something that really could make a difference. And the campaign at the moment is to try and influence MPs to put pressure on ministers and the government, whichever party your MP comes from. It's not party political. So with that, this is my last slide. I'll put up links on that link tree. I wrote a book, a very short introduction, just 100 pages on animal behavior. There's my pheromones book. There's a talk, a discussion in our time of myself, Jane Hurst, and Francis Ratnieks talking about bees, all about pheromones, and there's different animals. And there's the campaign for the National Emergency Briefing, which is something that you can join very easily online. So with that, thank you very much.
Thank you. That was super interesting. So I think we'll start with some questions now from the audience. So I know you've just talked about climate change, but we do have already one question about it from Zara. So if you'd like to add anything else to it, because Zara asks, what is the effect on climate change on the release and marking of pheromones?
That's a really good question and understudied. What we do know is things like those hyena marks have evolved for particular temperature profiles in the savannah. And if it gets hotter, those volatile molecules are going to disappear sooner. And that's also true of those mouse markings that the house mouse leave. So what we can say is that in rather unpredictable ways, except for those volatile molecules going to disappear sooner, we are probably going to have some disappearing signals, possibly some community breakdown. And I think what's really challenging is we know really generally so little about the behavior in the wild and how these pheromone signals work. To look at the changes is yet another thing that is simply unknown. But really good question. Thank you.
Great. I don't think we've got any other questions yet, but if anyone's got any, just type them in the chat or in the Q&A box at the bottom of the screen, and give a couple minutes just to see if any questions come in. But otherwise, I will send around the recording of this webinar to everyone in case they want to rewatch it. And also, if you've got any links to your book or to your other work, Tristram, then I can include that in the email I'll send out to everyone so they can easily access that. Okay, we've got a few more questions coming in. Another one from Zara. Do you have plans for future studies?
At the moment, I'm focused on activism. What led me to activism was starting the third edition and starting to realize that so many of the more interesting animals would be gone in the lifetime of the students that would be reading the book. Now, house mice and cats and dogs, they are going to be with us for sure. But elephant shrews and all sorts of other animals, those mara, many of those were threatened. And so what I saw as a priority was trying to do something to save habitats and act, or rather, try to persuade the people with the levers of power to act on our behalf to stop the changes that are putting so many animals in danger. I'm still fascinated, though, and there are some lovely studies coming out of Japan, for example, about all those responses to catnip. And just to talk about that for a moment, some of the early signs suggest that that cat response is actually cats anointing themselves with these smelly molecules that are produced by the catnip plant and a few other plant species. And these chase away mosquitoes. Now, I didn't know that mosquitoes target cats, but apparently they do get bitten. And these molecules that are self-medicated, as it were, onto the fur of the cat by rolling in the catnip are sufficient to send them away. So there are all sorts of interesting stories coming out. I don't have a lot of patience. It takes a long time to do experiments. So I'm just fascinated by what other people can find. And my role is synthesizing and drawing those stories together. But thank you.
That's a very interesting anecdote about the cat. Um, Estelle asks, in terms of species that can hybridize, for example, the wild cat and the domestic cat, do you think that can contribute to species decline? For example, could, yeah, sorry, I can't really make sense of the last bit of the question, but I think you get the
Yeah.
idea.
And the answer is yes. And I suppose what that is a sign of is recently diverged species or proto-species. And that's one of the things, for example, that's looked at across these hybrid zones in Europe with closely related mouse subspecies. What you sometimes find is in those hybrid zones, you get selection for difference. So you'll actually find that the smells that the species produce where both species are present, or both subvarieties are subspecies are present, start to vary. And the reason for that is that there's sometimes, depending on what stage in speciation, there's actually a genetic cost to mating with the wrong mate. And so what you'll gradually get is a selection for divergence in the signal. You're right. In the case of the wildcat and domestic cat, we're not at a stage where there is sufficient difference to avoid that interbreeding. And if we get interbreeding, then the differences that do exist disappear because the genes are all mixed up again. So yes, that is a problem.
Great. We've got a few more now coming in. We've got probably about 7 minutes left. Probably got to wrap up, so I'll try and get through these. Sarah asks, to your knowledge, is there much work that looks into bioacoustics and pheromone concentration production? Which species or taxa is this most known in?
Ooh, really good question. Uh, one of the pheromone researchers in the States actually has been looking at ultrasound, ultrasound vocalizations in male mice in response to pheromones. So your question is spot on. And that's a very good point that mammals do have multimodal signals. And what we tend to do as scientists is try to study one thing at a time. And as that mouse example showed, if you switch on the ultrasound microphones and listen carefully, you discover that actually um, mice can, as it were, chew gum and scent mark at the same time. So yes, there are lots of examples of animals that are doing both, and I'm sure that's true in elephants too. But as you go across the animal kingdom, in mammals in particular, you'll find that animals are combining signals, even if scientists don't always realize that.
Great, thank you. Someone asks, would you have any advice for students in animal behavior or animal science?
Always be aware of what animals are doing. You may spot something for the first time. Uh, Join something like the Mammal Society and local groups. There's always strength in learning from other people and other enthusiasts. Volunteer at a local zoo. It's full of smells and lots of things to observe. And you might be the first person to see something. And don't be discouraged. There's lots of interesting things out there, just waiting to be found.
Thank you. And someone else also would like advice, more specifically for researchers starting in animal behavior or animal sense. And are there any traps that we should be aware of to avoid making bad science?
What I'm really impressed by is how psychologists have responded to what was called by them the reproducibility crisis. So Ed Yong, the science journalist, said, the only reliable thing you can say about psychology is that you'll only be able to repeat the experiment reliably half the time. Most of the time it's junk. And in response, psychologists have started doing experiments more than double blind. They say before they start the experiments how they're going to do them and how they're going to analyze them. And so that's called um, registering, pre-registering your experiments. And it's something that's really taken off in psychology. And I and other people have been advocating that in chemical ecology and animal behavior in general, we should be doing that so that we can make our studies more reliable. And what really surprised me when I looked into this is even if you think you're doing it double blind, when you come to analyze the results, we always want an experiment to work. Well, speaking personally, you just get excited by the idea and you want your hypothesis to be true. The only way of preventing yourself from analyzing it in a way that will give you the result you want is to say ahead of time, before you collect the data, how you're going to analyze it. And that's a growing movement of pre-registering and even registered reports. And this is the last bit of this. It's taking off in animal behavior. If you're a research student, if you're an early career researcher, look into this, and my paper in 2020 gives an introduction to the idea. Basically, there are some journals now that will allow you to send in your proposed experiment. They will send it out to experts, get the feedback, and they guarantee to publish your paper, whether it's exciting or not, whether it gives significant results or not, so long as you follow the protocol that's been agreed. So if you're an early career researcher, you're kind of guaranteed a paper. And this could change the way that we do science. It makes it much more reliable. And I think it might be the way of the future. You can still do exploratory, exciting research where you've got no idea what you're doing. But when you try to test a hypothesis, you say what you're going to do before you start. You don't pretend after you've done some exploring that that's what you were looking for. So that's been likened to, if you do it that way, it's a bit like firing an arrow and then drawing a circle around it afterwards. So pre-registering your experiments and these registered reports mean that you say what you're going to do before you start, and that's a much more reliable way of doing science. And it's transforming psychology, and I think it'll transform animal behavior too.
Amazing. Thank you. All right, we've got 2 minutes left. Let's try and do two more questions. Maria asks, can you explain a bit about the physiology of chemical receptors on the vomeronasal organ of mammals? I probably completely butchered that pronunciation.
Not at
Do
all.
they work in a similar way to the olfactory receptors?
So olfaction was the last of the senses to be properly explained. And the Nobel Prize went to Richard Axel and Linda Buck only in what, 2003? So that's many decades after the Nobel Prizes for sight and hearing. And receptors are very like drug receptors, like the serotonin receptor. They occur on nerves in your nose. The vomeronasal receptors are work in the same kind of way, but they come from different families of membrane-based receptors. But they're basically doing the same thing. And what's amazing is in mice, they have about 1,000 of these different olfactory receptors covering an enormous range of molecules. They're sensitive to such a different range of molecules. As humans, we have about 400 that still work. Each of us has a slightly different working set, which means that when we're smelling cheese or sniffing coffee or wine, we actually don't necessarily smell it in the same way as our brothers or sisters or our friends. And it amazes me that we actually agree on what to eat at all. So if you think of color blindness with just three color genes, the color code genes, red, green, and blue, um, we have all different kinds of color blindnesses, about 9 or so at least. Think about the different ways that if you've got 400 different odor receptors in our noses, how those can vary. And that's why some of us like coriander leaf or cilantro, if you're from North America, and some of us don't. About 10% of the world's population, and it doesn't matter where you live, about 10% of the world's population doesn't like that green herb put into a curry or sprinkled on top. So that's one of those variations, and that's all down to the particular sequence of that particular receptor. So there's a whole growing literature about receptors. Similarly, there are some flowers that some people can smell and some people can't. And there's nothing you can do about it. If you're born that way, that's how it stays.
Great. And lastly, one more question from Bob. What research are you aware of in pheromones in aquatic species?
Now, that's a really important question. That's what I forgot to talk about altogether. And the answer is whales and dolphins and um, pinnipeds, so seals and walrus and carnivores that have gone back into the sea, actually have a very reduced olfactory repertoire. So instead of 1,400 or whatever olfactory receptors, they're down in the tens or smaller, different receptors. Now, I think there's a bit of evidence that some whales can smell. There are some odors given off by some of their prey that get caught up in the wind if there are big collections of plankton where fish are going to congregate on the animals that eat the plankton. Um, but carbon disulfide actually, I think, is the molecule there. There's actually no evidence that they have pheromones. There's a hint because some of the toothed whales have the Harderian gland, which I think our guinea pigs have, that might be producing some pheromones. Because if they did have pheromones, they'd only need that receptor. So they wouldn't need to smell more generally. But the answer is, we really don't know. But the evidence so far is that aquatic mammals probably don't use pheromones. And that's a very different story from fish. Goldfish have these amazing chemical duets, and they're exquisitely sensitive to the molecules given off in the urine. And they have these duets with molecules in the urine of the female, and then the male squirts, and she responds. Just a fantastic story. Whales and dolphins, probably not.
Great. Well, thank you so much. This is a really interesting talk. I hope everyone enjoyed it. Yeah, I'll send around the recording and any links you send me, Tristram, and I can also send those around to your book, etc. But thank you everyone for coming today. I hope you enjoyed it. And yeah, have a good rest of your day, everyone.
Great.
All
Thank
right.
you.
Thanks.
Bye.
Bye.